Yeah, good morning, afternoon, evening, middle of the night, early morning. So thanks a lot for the opportunity to speak. I will give a quick overview, when I'll also get this working, about what we are doing in Genomics England, particularly in light of GA4GH standards. And I will do so not so much from the perspective of as a researcher, I would like us to federate and share data, but rather from like an engineering and a products perspective of why these standards are helpful. And for those of you who were in the previous forum earlier this year, it's not dissimilar to that. So in Genomics England, we have two main aims. We are there to help evolve the genomic healthcare in England and wider in the UK. And we do that primarily by serving the NHS as a diagnostics facility where we have requests for a analysis based on whole genome sequencing for patients with rare diseases and cancer. And we provide the full stack all the way back to results going to the labs. And then as we are accumulating those results subject to consent, those participants' data, then we use that to fuel research by taking those data and presenting them to users, academic and from pharmaceutical companies, startups and so on, to be able to make discoveries that in turn flow back into the improving health. And indeed, we have a number of examples, hundreds, where research has led to new diagnosis in the NHS. So this very now common paradigm of its infinity loop is precisely how we see our role in the ecosystem. Good. So if I focus on that first healthcare loop, briefly, what we do is we receive a test order from the NHS. We sequence data through Illumina technology that comes to us, and we do alignment and variant calling. We do so by orchestrating a number of pipelines and storing some data. Once we have those alignments and variant calls, we do the standard that scholars have presented before around variant interpretation, annotation, prioritization, those sorts of things. And we do so by applying a number of what we call interpretation services. And for example, this pharmacogenetic analysis that Mark was just speaking about is one of those additional findings, but there's also ways of prioritizing and classifying variants in rare diseases and cancer. And finally, we make that data available for the labs on the right-hand side. We call them genomic laboratory hubs in the new genomic medicine services in England. And those labs have access to those data, and they do various things. As you will be familiar, they would browse those genomes, perhaps through a standard genome browser like IGV. I'll show you an example in a second. Or they will review the variants and the clinical information and the scoring of those variants to be able to diagnose those cases. And finally, those data are deposited in variant knowledge bases. So as we're doing all of this, there's a number of challenges, and we have started to implement more and more GA4GH standards to solve those challenges. For a while now, we've been using CRAM as our data storage, and that essentially saves money, but also the tool chains now to run those pipelines and so on are by and large fully compatible with CRAM. So there's no need to inflate and deflate from CRAM to BAM and so on. So that's been very helpful. The one where we are perhaps most mature of all the other standards is HTSGET, and I'll just in a second present a slide on that. So I'll leave that for a second. But it is our primary way of communicating genomic data to our network of laboratories. One of the big problems we've had in the way we run pipelines is that there are teams that are doing R&D trying to establish new pipelines for cancer, for rare diseases, for cancers that have high contamination in the normal from the tumor, those sorts of things. But the process of taking those pipelines and then bringing into a production system has been quite complicated and tedious. So we have started as proof of concept, as I say here, seeing how we can use just the standard workflow execution services from GA4GH standards and adopting tools such as CRAM that was mentioned earlier to match both what would be the ISO created to clinical standards pipelines, but be able to flow in the R&D quicker into it. The same case here, where we this is slightly simpler services, so we're using the task execution service, and it helps in the same ways. Very similar story here. If we move into the data space, we have the data repository services. One of the big problems we have is we have data both in our data centers, but also on the cloud. And it means that mounting directories and knowing what file is where and what do I have to, how do I access it, has become a serious hindrance. And therefore adopting this standard is very helpful so that we can have a single way of identifying a file in the world, regardless of whether it sits on-prem or on the cloud or in someone else's cloud. If we move forward now into the future, then we have this very knowledge bases, and we heard yesterday from the ClinVar folk that that they are starting to open APIs for submission from their knowledge bases. So this is an area where we are participating quite actively, both through GA4GH, but also from an implementation perspective, seeing how we can use those standards to represent variants better. And finally, we strongly believe that we need to share that information across the world. And so things like Beacon2, where it's not just do you have this variant, but can have a lot more information about that variant, something that we're involved in right now, certainly in the scoping and following closely, but we haven't started to consider the implementation. So this is, I hope, gives you an idea of us, how we're using those various GA4GH standards, not for the great ideas of federation and data sharing, but really just to solve some of the engineering problems that we have at present. Let me spend just a second on HTSGET in particular. So for us, Genomics England holds all the CRAMs and the VCFs for the genomic medicine service when it comes to whole genome sequence. But the labs, as you'll be familiar, quite often need to look at those variants and look at the read level support to ensure that they are comfortable reporting that variant eventually to a patient. And so the previous ways we had to disseminate that data were quite clunky. And once HTSGET came across as a standard, we decided to go ahead and use that, and we created an implementation that is relatively well performing. It's open source, and you're more than welcome to give it a try if you're desperately looking for an implementation for HTSGET for your own solutions. So let me give you kind of an idea of how that is. Here is just showing you the, if you're familiar, just the API call, HTSGET, where you can give what formats and the positions, and it will, with authorization and authentication, it will return the data. And you can use that in a number of ways. Here is simply calling it from some tools, where you can provide nowadays, because the toolchain supports HTSGET, you can just provide the HTSGET URL, and you can see here the resulting BAM, or the CRAM, I mean, and you're exporting that as a BAM. But you can also load it into a browser. And so here is the rather clunky browser that we have. Is there based on IGV, but you can also select files that you want in there. And it's just showing you from a lab perspective, being able to get data centrally across the country. That's been quite a lot of success, and it really has solved a problem that before we had a number of alternative standards. The second one is more from the research loop. So in this case, we have a research environment where our data, and by our data, I really mean the data from the participants, and we are custodian of that, but just shorthand for it, is read-only, so you cannot download the data from the research environment. That is the commitment we've made to our participants so that their data is kept safe. And so that's a pretty high barrier for people to just come and use it. People are normally, users are normally expecting the institutional HPCs or the desktops. You download the data, you have all the tools, you're familiar with those environments. And so the question is, how do I bring my data? In this particular case, how do I link a repository, or how do I bring my own workflows, and that's that task repository service, and how do I execute those workflows, and that's workflow execution service and task execution service, particularly without the need to go through a steep learning curve to learn yet another, here's GEL's new environment, or here's UK Biobank's environment, and so on and so forth. I don't want to spend a lot of time trying to learn, I just want to use the data. And so that means that for us, we can have more users and tell the government that we have lots of users writing lots of great papers, and please keep funding us. So just from like a simple perspective of getting the data used, that's fantastic. But the alternative is you have to learn another workflow execution service. Perhaps you have to become either familiar with Nextflow or WDL. Not everyone is familiar with those. You have to learn how to use GitHub very well so you can bring code in. Same with S3 to bring data and so on. So this service just provide a single standardized way for the community to just go ahead and use them. And yeah, I made the point here that actually it's a federation in the case that to bring all these data sets, to come and use it together, rather than a lofty goal, is really just a way of achieving our mission of getting the data used. So one of the kind of most exciting things that I think I find in this space is the very simple what's happening in the cloud workstream in GA4GH. And here's just an example. This is Docker store where various the workflows are maintained. This particular one is WDL. And the point is that if I have the right credentials, and there's another workstream on that, then I can go ahead and launch this tool in any of those ones. And what we are working towards is to add something here that is a GEL trusted research environment. But it means that as a user, I can have easy access to the tools, I can bring in more tools, and I can deploy it across a large federation of repositories. And we'll hear, I'm sure, much more about this for those of you who are going to be at the GA4GH meeting next week. Great. So in summary, I would just like to emphasize the point that GA4GH standards are not just there for genomic research, but it's also there for clinical standards. And some of them are ready for production systems. HTSGET works really well, WES and TESS, they're used by many people, as well as the authorization, authentication, passports are becoming quite robust. One of the big problems, though, is that the implementation is challenging, and so implementing those such that they're performant at the scales that we need them to be is difficult. And so some of the cloud vendors are doing some of that work, but not everyone is on the cloud. So I encourage us to share them, because if we don't share those standards, we're just not big enough as an ecosystem to have many different implementations, people coming to them. We need to share them and not reproduce too much work. And then the last one is just a call. There are many evolving standards, and we've heard about some yesterday, packets and pedigrees, but there's also Beacon 2, ways of thinking about interpretation and evidence reporting. And they're only going to be right and therefore useful if we are involved in participating, at least providing feedback, but ideally in those communications. So it's a call then to be part of that, so that we can all one day be able to interpret in better ways for our patients. Thank you very much.